And then the second thing you know about ARVO, right? So these are the two introductions for Sugo. So he was trained at NEI in genetics, human genetics, and then he uses that for his research as well as in the clinical world. And then for the ARVO, so anybody who contributes heavily to the organization, he gets a fellow, recognition as fellow of ARVO. And he is a fellow of ARVO, and he will be inducted as the golden, right? So this year. So those with that introduction, and then the other introduction is, of course, I inherit one of his student here as my postdoc. So that's another relationship I have with Sugo besides all the personal relationship. But let's welcome Dr. Chakraborty for his talk, on glaucoma genetics. Thank you, Sugo, for visiting UW.
Thank you, Dr. Patnaik, for that warm introduction. And yeah, indeed, it has been a pleasure interacting with you over these years, and we have also gained from your insights. So what I'll be talking about is a disease that is largely seen in the developing world, and not so much so in this part of the world. And the disease is primary congenital glaucoma. It's a glaucoma in children, occurs almost at birth to within the early infancy. And we have been trying to kind of understand the mechanisms of what happens in primary congenital glaucoma and what are the insights that we get from evidences in molecular biology and human evolution. So as I mentioned, that it is a severe form of defect in children, and largely due to the developmental defects that occur in the trabecular meshwork and anterior chamber angle. As you see, in the Western world, it is not a major problem. It's around 1 in 10,000 to 1 in 30,000 live births, as opposed to if you see populations in the Middle East or among the gypsies, or even in India, the southern part of India from where we come from, it's around 1 in 3,300 live births, contributing to over 4% blindness in children. So that's the magnitude of the problem. And our aim over the last 2 decades has been to develop a global blueprint for completely understanding this disease, more from a genomics perspective. So just to give a small overview, that what we are trying to understand here is rare alleles. In other words, we are trying to understand mutations which have a very large effect and are very rare in the population. And at the same time, we are trying to identify those mutations, characterize them, understand their functions, and link them to the phenotype in order to do a genotype-phenotype correlation that may be translated back into the clinic in order to understand the effects of those changes in terms of clinical management and prognosis. But with the advent of the genome, we know that there are other variants, the intermediate low frequency variants and common variants also, that interacts in any disease conditions. And primary congenital glaucoma, I'll refer it to as PCG, hasn't been averse to that. Even this, which was supposed to be a Mendelian disease, is behaving more in a complex manner. So in terms of genes that are involved in different forms of glaucoma, and by the way, genes overlap in terms of congenital and other forms of glaucoma. So what we are trying to do is not only look into genes which harbor mutations, which we call as candidate genes, like the first gene that was mapped was a cytochrome P450 CYP1B1, and globally there are a large number of mutations, and followed by other genes like the LTBP2, the TEC, MYOC, and FOXY1. And your chair, Terry, was actually instrumental in mapping the TEC gene. So there are other genes of intermediate and effects and common variations that interacts with these genes, and their functions are what makes the condition more complicated. So if you look into the profiling of the candidate genes, if you see CYP1B1 across the world, so the majority of the mutations are again in the Saudi Arabian populations in terms of frequency, and among the Slovakian gypsies. In the Indian population, we see close to around 40%, but these are just the mutations. These are not necessarily homozygous mutations, because we're talking about a condition which we see more in a recessive form. That means you need two disease alleles to manifest a phenotype. So in terms of the homozygous allele, the frequency would come down faster, would come down to a much lesser extent. LTBP2 and TEC are relatively less common in the Indian scenario. And as Bikash mentioned, Meha was also instrumental in identifying many of these mutations in the Indian scenario in a large cohort of cases. But initially, we wanted to understand why is it that it's only in CYP1B1 that we get a proper profiling of these cases across multiple populations. We went on to do some or gain some evolutionary insights, and we wanted to see that are there signatures in the genome that harbor or that kind of facilitate these mutations. And we find that if we take intragenic variations in these CYP1B1 gene, there are specific SNPs that are kind of consistent across populations. And in order to understand that, we looked back into the great apes. And for instance, in chimpanzees, we find that these SNPs are just one mutation step away from the human genotype. So there are single step mutations, and the mutation that or the haplotype that is constructed out of these are favorable because these have been fixed, or these were the founder haplotypes when one sees the evolution from the great apes to humans. So the CGGTA haplotype, for instance, is the most common haplotype that we find in cases. So to cut a long story short, no matter which population you come from, let us say if you harbor this G61E mutation, whether you are from Ecuador, Morocco, Saudi Arabia, or India, you are on the same haplotype. And likewise for the common mutations, they are harbored on the same haplotype. Likewise, there are a few recombination derivatives, as you can see, like the CGGTA, and there is a single step change to CC, and that harbors a different type of mutations. So globally, these mutations are structured both in terms of geography and in terms of their migration, and we could actually trace it from Africa to further southeast. The reason that some of these mutations are more prevalent in populations of Africa or Saudi Arabia or gypsies is because they have a founder mutation. And as populations migrated out of Africa into Asia, into the Far East, and to Australia, you have a lot of recombinations occurring under these haplotypes. And that might be unfavorable for the mutations. And so if you see populations from Africa, Saudi Arabia, India, and compare them to Southeast Asian populations or Far East Asian populations, you see the frequency of mutations reducing drastically, and that goes in line with this, with the haplotypes. But this is true only for CYP1B1, again, because CYP1B1 has been found widely across all these populations. We tried to do a genotype-phenotype correlations based on this, and we find that if we take single mutations or single allele, they are not sufficient enough to explain the correlation with a particular phenotype. We tried to do it with presenting intraocular pressure, corneal diameter, cup-to-disc ratio. If you see across cases harboring mutations at the pre-operative stages and further follow-up following surgery and over a period of time, what you find is that cases with homozygous, heterozygous, or cases with no mutation do not really show any difference in terms of genotype-phenotype correlations. And we also kind of replicated this along with other populations, and we had a large replication cohort from Brazil and India, and the story is almost similar. But what we did find is that there is a variation in the CYP1B1 promoter, which lies between the Tata box and the DNA response element, and that changes the genotype from T to C, and those individuals who harbor C have a reduced CYP1B1 activity, right? And interestingly, this promoter variation is strongly linked in terms of linkage disequilibrium. It's strongly associated with a coding region mutation. So individuals who have a coding region mutation and a promoter change ultimately have a reduced CYP1B1 activity. Further, we showed that this also leads to defective binding of other transcription factors in development. Now, clinically, if you follow these kids post-surgically, you see that those who harbor those mutations as well as the promoter variation have a poor prognosis in terms of their IOP control, as opposed to those who do not harbor over a period of time. So that was one part of the story, and then we moved on to, because that, as I said, CYP1B1 by itself, if you take homozygous mutations, it's only around 20 to 25%. So what happens to the remaining cases? We tried to find out through runs of homozygosity across the genome, and we found that when we kind of scan the entire genome over a period of across all the chromosomes, there are few regions in certain chromosomes which harbors homozygosity. But at the same time, that doesn't explain the overall mutation profile that we get in CYP1B1. We did similar exercises with LTBP2 and didn't find it. So at this point of time, we had to slightly make a change in our strategy. And the strategy was to kind of initially do a targeted sequencing. So you select genes other than CYP1B1 that might be contributing to PCG. And these genes, as I said, PCG is one form of the disease among developmental glaucoma, which is very classical, but there are other forms of developmental glaucoma as well, which share certain phenotypic or clinical resemblances. Interestingly, there are genes that kind of overlap across these phenotypes. For instance, anterior segment dysgenesis genes and other glaucoma genes. So we prepared a very large targeted panel of 352 targets, did targeted resequencing, and what we found was interesting. The first thing we found was the gene that was identified by Terry using whole exome sequencing. We could also kind of map it through our targeted sequencing. But the differences from the American patients to Indian patients was that these mutations were heterozygous in our case. Similar it was in the US patients. But in our cases, we found that they were interacting in a digenic fashion. And this large part of this work, again, as you can see, was done by Meha. So what was found is that the parents are either heterozygous for a CYP1B1 mutation or a TEC mutation, and they are asymptomatic. The child, when inherits both the heterozygous mutation, they show the phenotype. And there is an interaction. So this is a genetic interaction. We further showed that there is a corresponding physical interaction. So when we express this in HEC3 cell line, what we find is that the wild type interacts. But for these mutations, when you look into their interactions between CYP1B1 and the TEC gene, you see that there is a perturbed interaction. The interactions reduces. We could quantify and show that there is a reduction of these interactions. And further we showed in endothelial cell lines that based on the mutations, if the mutations are intracellular or extracellular, there is a different cell migration of those when we see with respect to the angiopoietin 1, which is a ligand of TEC. So if you look into the ligand angiopoietin, you see that intracellular mutations tend to migrate outside the nucleus, and extracellular, they are more confined. Clinically, also we found that these patients who harbor both mutations in CYP1B1 and TEC, since there is a perturbed interaction, perturbed physical interaction, they also have a severe phenotype in terms of prognosis. And we found that across other genes also, like for instance, with other glaucoma-associated genes, you find, like in SEP, for instance, you find similar physical interactions, which often are perturbed. We then looked into other genes. One of these was a serine protease, 56. This actually is again involved in development. At the same time, it leads to other anterior segment phenotypes. But what we found that PRSS56 harbors mutations, specific mutations that are only seen in primary congenital glaucoma. And as you can see, these are highly conserved across species. And some of these mutations also interact genetically with CYP1B1 mutations. In collaboration with Simon John's lab, we showed that PRSS56 mice developed intraocular pressure, and there was a structural change in terms of the shifting of the Schlemm's canal and the trabecular meshwork in the mutant as opposed to the wild type. And we also find that PRSS56 does not necessarily interact with the other glaucoma-associated genes. It's more isolated. And what it means is that there is a progressive degeneration of the trabecular meshwork in the knockout, where you find that the TM almost becomes hypoplastic as opposed to the wild type. And when you rescue this phenotype with EGR1, which is the early growth response protein 1, you can rescue the phenotype, but the intraocular pressure still remains high. So that means intraocular pressure by itself has a different pathway altogether, or the genes contributing to intraocular pressure may not necessarily be those which contribute to the structural defect in primary congenital glaucoma, but since primary congenital glaucoma again is a developmental disease. So this was there in development and mechanisms. Recently we also found that GLIS1, this is a glycemilar gene, is also involved in glaucoma pathogenesis. And using samples both from the JERA and the UK Biobank cohort, we could map a unique variant in SNP1 in GLIS1 that actually is associated with primary open-angle glaucoma. We also correspondingly showed that GLIS1 harbors specific mutations, again in CYP1B1, and again those are highly conserved mutations. And what GLIS1 does, you also see that GLIS1 knockout also kind of has a progressive degeneration of the trabecular meshwork. And we also showed that they have a relatively high intraocular pressure. The knockout has high intraocular pressure. If you take the contralateral eye and you use anti-glaucoma medication like ripacidil, it again resembles a wild type, but on the other hand, the other eye in the knockout also has raised intraocular pressure. Then we did some chip sequencing and also found that the genes that interact with GLIS1 are those which are expressed in the trabecular meshwork. So obviously GLIS1 might be playing a pivotal role again in contributing to glaucoma pathogenesis. This was, we reported these results last year. So what does all this story tell us? It tells us that across multiple alleles, across multiple genes within our targeted sequencing panel, we found a large number of variants. We also found low frequency and intermediate variants, maybe of relatively lesser frequency, but obviously there are interactions both at the genetic level and in some cases which we proved that there are corresponding physical interactions also for those variants. These rare pathogenic variants are distributed largely in the cases. You rarely find them in the normal control. So we wanted to see if it is a phenomenon only restricted to a local population, but we also looked into the 1,000 genome databases, our own large cohort of controls of close to 2,000 individuals. We actually subdivided the 1,000 genome individuals into those who are ethnically related, for instance, Bengalis from Bangladesh, Punjabis from Pakistan, Gujaratis from Houston, Sri Lankans from UK, etc. And we also find that across those, the profile is almost the same. These are very rare variants, hardly found in normal individuals. So 62 such pathogenic variants were observed. These were different multiple genes and these rare variants. And additionally, as I said, in our controls, we do not see their representation across any of these phenotypes, across any of these normal controls. And when I say normal controls, these are controls which we have enrolled from an epidemiological cohort, which we have been following up for the last 20 years. So for the last 20 years, we have been following a cohort within the region, which is ethnically matched, and they have been examined clinically for all the parameters. So at stage 0, at stage 1, and stage 2, and they do not show any ocular problems. So these are more ideal controls for our cases because we are looking into germline changes. We also found there are significant haplotypes, signatures that are there across these genes which were found. And then that leads to more complexity. And the complexity is that we established that, we and others have established that not a single gene is enough to contribute to a phenotype. We thought, okay, it might be 2 genes where we show genetic and physical interactions. But now with the complete scanning of the genome, what you find is that these are the cases and these are the genes. So there are multiple levels of interactions in each case. For instance, if you see, these are differently coded homozygous mutations, compound heterozygous mutations, and heterozygous mutations. If you see across individual cases, it may be interaction of a homozygous and heterozygous. It may be interaction of 2 heterozygous or 2 or 4 heterozygous mutations. So one really needs to tease out of these which are the modifier alleles. So that is one of the challenges which we have, and I think everyone in the world has the same issue, to kind of delineate which are the potential modifiers and which are the causal variants. So that is where our work is geared at. And in terms of the common variants, again, you see that there is a profile across different genes. So if you see with respect to glaucoma-associated genes, there are variants which confer risk, some which confer protection. Again, with anterior segment disease-related genes, you see that there are risk alleles and protective alleles. Now, all of these together interact. So these are only for the rare alleles, but the table would be much larger when we bring in the common variants. We tried to understand it from a network point of view, and we did some pathway analysis of this. And what we find is that most of these pathways are involved in similar molecular functions, both in terms of transport, migration, cell-to-cell adhesion, organ development, and also the post-translational modifications. And the same thing applies for anterior segment disease genes as well. We also looked into other genes that harbor mutations, like for instance the crystallins. Again, these are involved in development, certain sections of crystallins. And we found that even crystallins harbor mutations in congenital glaucoma. And these are very specific mutations which are kind of highlighted, which you do not see in other congenital cataract phenotype, for instance. So again, these ones also lead to different levels of interactions. You see that they interact not only with CYP1B1, they interact with a whole lot of other genes. And this is another area which we are now currently trying to dissect. In one of the ARVO meetings, this was a poster that was presented some time back, and Dr. Patnaik had suggested that why don't you do some ERGs, why don't you test for the ERGs in these patients? And a suggestion was that can you actually see a profile of photopic negative response, for instance. And we started doing that, so across patients and their families. And what we find is for certain parameters, certain PHNR parameters, indeed there is a kind of an association. We still need to dissect it. We'll discuss it more over time, like for PHNR at minimum resolution, minimum milliseconds. There are certain associations when you categorize cases harboring mild or severe phenotypes, and within those phenotypes, let us say if there are similar set of genes, you still find a variable response that are statistically significant. So this is another part of the data that we are working on. So the last part, what we wanted to do along with is to identify the genes, identify other cases that do not harbor mutations either in candidate or the targeted gene panel. We wanted to do a whole exome sequencing, and then our approach was to use trios, that is the parents and the affected child. And since most of our cases were consanguineous, we took this to our advantage to identify homozygous mutations. We did find there are mutations which are specific to these cases, and some of these are also replicating across unrelated families. So those might be essential targets. This is very new data, and why this is important is because they are expressed adequately not only in the fetal tissues but also in other ocular tissues. And obviously they are involved with the regulation of cell death, with blood vessel morphogenesis, vascular development, and so forth. So in our large cohort of PCG, we found that if we take only candidate genes, that is only the 3 major candidate genes, we have close to 58% which is unknown, right? When we use the targeted panel, that we have another 10% that is contributed by the other genes. When we use whole exome sequencing, we reduce the number of unknowns further. And these are only with respect to individual genes that harbor mutations. These are not with respect to genes which show interactions, because again, that's a different algorithm, and we are trying to find out that. But why these genes are important is because of their GERB score, that is, in terms of their evolutionary potential, the ones that are conserved with respect to their sites as opposed to those which do not, which harbors the common changes, they do not show a high GERB score. So we are now classifying them and then trying to figure out what levels of functional interactions exist and what should be the study designs for understanding functional interactions across multiple genes to find out which is the modifier allele. So based on the data what I presented, we have genes which are leading to developmental defects, genes that are leading to cell death, and finally contributing to anterior chamber angle defects and trabecular meshwork defects leading to primary congenital glaucoma. So again, our proof of concept, apart from the functional assays, is that we are using this, as I said, from a large cohort of, a large epidemiological cohort, and we see that these variants have a very low population attributable risk. By itself, a single variant does not contribute to the disease, but using that strategy, we are also, that's a translational angle that we are kind of taking on now, to develop some risk stratified treatment in the population if we are able to find out which are those alleles that confer highest susceptibility. So our understanding of pathogenesis is based both on the genetic interactions, the physical interactions, the characterizations, and now we are trying to integrate all of this to understand causality. So finally, since we are a clinical institute, we would like to lead from genotype to phenotype. So understanding a more structured phenotype, which will kind of allow you to understand what are the genetic components that might be useful for the diagnosis and the prognosis for a better clinical management. So to conclude, we showed that globally mutations in PCG-associated candidate genes are structured by geographic and haplotype backgrounds, and there are evolutionary implications. We showed that combinations of exome and targeted sequencings, they reveal the functional involvement of genes with varying magnitudes of effect in PCG. And ocular angle development and raised IOP are complex mechanisms, and they are not intertwined. They have multifactorial etiology and functional interactions of genes involved in these pathways that maintains ocular angle structures, and IOP may hold key towards understanding PCG pathogenesis. So I'd like to thank all the patients and the normal volunteers who have kind of contributed to our efforts, and our team, currently the graduate students. You see Meha also, who moved on to be here, and the clinical collaborators from outside India, particularly Hemant, Deepak, Saida, Simon, Anton Jatin, and the funding agencies. And at this point, I'd like to stop here and take any questions. Thank you very much.
Thank you, Sugo. We can take questions from the audience. We can take a couple questions.
So my question is, when you're sort of looking for pathogenesis, you come across any other protective steps, ones that might also help us evaluate clinical impact.
Sorry, the second part.
And just that will help us evaluate the clinical impact of the different mutations.
No, so what we are trying to understand is in the total spectrum of things, like you have rare mutations, you have intermediate changes, and you have the SNPs, which are common variants. Genetically, we see their interaction. We are now trying to dissect what will be the best approach to understand them functionally. And because these are common variants, that makes the situation more complicated, because everyone, a general population, many individuals in the general population also harbors the same. So to what extent the combination of that along with this leads to pathogenesis is what we are trying to get at. So we could see with rare mutations, like I showed you, both using in vitro and in vivo approaches, we could show with rare mutations. But there is also association of others which cannot be discounted. So when they need to be taken up into the scheme of things, and we are trying to find out what will be the best approach. So one of the approaches could be that if there are multiple mutations, the rare mutations, you could kind of do this CRISPR knockouts and then try to see the effects at the phenotype. But these are not one, as I showed you in that scheme. There are multiple genes. So first we need to kind of tease out which are the potential modifiers. Can we get it based on our comparison with the controls or with the bioinformatic assessment, and then take it down to functions.
So any other questions here?
Yeah, I came late, so if you address this, I apologize. So have you considered the epigenetic aspect of your data? Like sometimes the SNPs could actually change the CG island, and that can result in a difference in the methylation.
Sorry, I didn't get.
CG island, so sometimes some of your mutations could actually
Right,
change
right.
the methylation status of a lot of genes.
Right.
And another thing is actually the microRNA. I don't know whether you have looked at that or not.
No, we haven't looked at microRNA, but we are definitely looking into the epigenetic regulators, both the CPG islands. That data is not yet complete, so we are still, that's a work in progress. Yeah.
Again,
But at the
thank you.
same time, we are also looking into the tissue morphology. For instance, but again, the challenge is that from these babies, you get a very minute amount of tissues, and to culture it is again another challenge. So we are getting this trabecular meshwork. We are trying to do a morphological assessment initially, and then our next approach would be to do some kind of a spatial sequencing using the single cell approach to see are there different profiles across those who harbor mutations versus those who do not.
I don't know.
Yeah.
So that's actually very interesting. So I would suggest, a lot of the microRNA are actually located in the introns.
Right.
And some of your mutations may not be actually picked out if you are looking only at the candidate genes.
Right.
So, and you want maybe really to focus your analysis on the microRNA and to try to nail it down to
Right.
see whether, because some of those microRNA can be really a master regulator of a lot of that's,
Exactly. So we are going more from the gene side. So other than candidates, as I showed, our approach was initially to do with the candidates, then with the targeted genes, and then with whole exomes. So now another story that is coming up is also the mitochondria, because many of them have a crosstalk with the nuclear genome, many of the mitochondrial variants. So we are also trying to understand in terms of the mitochondrial activity that are there, kind of insights that helps us understand why those genes suddenly get upregulated in PCG. But yes, your suggestion is kind of well taken that the microRNA profiling would also be, but we haven't got started yet with that. Yeah. Yes, yes, yes.
Thank you, Subho. I have also some comments
Okay.
which I'll share with you later. But now we can get to the second part of the talk. So as you see, I have inherited his student. So inheritance is not just Mendelian, but you can also see non-Mendelian inheritances, right? So that's what you see. And that's what is the specialization of Indrajit, who is following Subho. So she is looking at both Mendelian as well as non-Mendelian inheritance using omics approach. And then I know, because they both work in genetics, so that's a common talk in family too, right? So after work also they talk about omics. But then it's not the same disease though, so they wanted to be separate that way. So she focuses more on ROP, right?
Yes.
And AMD. So we'll hear from Indrajit on her approaches, omics approaches to tease out some of the phenotypes that they find specifically in that subcontinent in terms of ROP and AMD. Thank you, Indrajit, for visiting us.
Thank you, Vikas, for this opportunity to share our work. So I'll introduce myself, as Vikas just mentioned. We are at LV Prasad Eye Institute, and my major focus, the focus of my lab, is primarily on the retinal vascular diseases such as AMD, diabetic retinopathy, and ROP. Today I'll be talking about the work on ROP. The primary interest in retinal vascular disease is this, that we see these diseases, they have common features, like they have problem with the vascular abnormalities in the retina, and they are diagnosed based on that and also treated for that by giving suitable anti-VEGFs or various other molecules. But I think the problem doesn't start only with the abnormal vasculature. That is only the result. What is happening is much beyond and much before vasculature, errors in the vasculature. That is probably is in the retina, and then in the there is neurodegeneration in the retina, which is not picked up by the current tools. And so by the time the phenotypic features develop, it's already that damage is already done. And so my lab is more interested in understanding the neurodegenerative changes in the retina, which could contribute to inflammation and vascular changes later in the eye, which are being looked at in more details everywhere. So with this introduction, I'll just start my talk on ROP. ROP is called retinopathy of prematurity. This is a condition which is exclusive to premature born babies. It's a quite important condition in countries, particularly like us, in Asian countries, where the prevalence of disease is, the incidence rate is very high. And this graph shows the global figures of ROP. So in a year, about 184,700 babies, premature born babies, develop some degree of ROP, and of these about 53,800 develop severe ROP, leading to long-term visual impairments. And that can, and there are no mechanisms to really treat the disease, except that you can stop the progression of the disease. But this disease is very interesting because it's understood that ROP is contributed largely by the immature vessel network formation, not just in the eye but also in the body, and that kinds of gives a kind of less oxygen supply, when the baby is born, and that leads to, this abnormal blood vessel proliferation in the retina. But the interesting part of this disease is that this disease has a spontaneous regression. So what we don't understand is why in some babies we see spontaneous regression of the disease, whereas in the other babies who are treated similarly or who were born at the same gestational age or birth weight, they do not they do not have spontaneous regression, and despite of all the treatment given to them, they go on to develop severe ROP, and eventually they lose vision. So this is the question that we have been chasing. As I said, in India about 40% of the premature born babies, they develop some form of ROP, and of them 18% of them develop severe ROP. This is a quite huge number. Our institute has actually developed a state-wise ROP screening program where the ophthalmologist and the fellows in our hospital, they are trained to go to all the neonatal units, and they examine all the babies who are born prematurely. And then if they see some kind of changes, or if they see there is immature retina, when we say immature retina, that means the blood vessel network is not formed, then those babies are followed more closely. And as in when we see there is some kind of vascular proliferation which is going in a normal way, then these babies are treated either by lasers or by cryotherapy or by giving anti-VEGFs. So the efficient management of this disease is highly dependent on both early diagnosis of which can and so we need markers that can help us in predicting the risk of ROP progression, and also a very timely and effective personalized treatment strategy. So toward this goal, so this is just the classification of ROP. So in a developed retina, you see a proper fully vascularized retina, but in ROP, in the initial stage, we see this is the white colored, this is the avascular retina, and this is the vascularized retina. And this demarcation between avascular and vascular retina deepens as the ROP progresses with the formation of a ridge, and in the subsequent stage, there is a proliferation of blood vessels from this ridge, which causes tension on the retina, and retina gets detached, and that's how the child loses the vision. So there are several risk factors which have been looked into for their association with ROP development. However, only gestational age and birth weight are the two factors that could actually show significant association, while rest of the other factors which have been listed, both infant related and maternal factors, do not show really any good association or predictive risk for ROP. So as I said, so our question was that how do we differentiate which are the babies who are at risk of developing ROP, and can we then look at some novel methods of targeting this disease. And toward this goal, we have used omics based approach, where we are looking at genomics, transcriptomics, proteomics, and metabolomic profiling of these babies, and to look for some understand what are the pathways that contribute to disease, and subsequently to develop newer drug targets. So when we began this work, we first actually looked at the genes that are involved in the retinal maturation and in the retinal blood vessel, the vasculature formation, and we did a targeted gene screening by using SNPs in these genes. And so we have picked up about 384 SNPs from 28 such genes which are involved in the pathways which are listed here, and then we typed them in about 188 patients and about 167 premature controls. So of these SNPs that we have targeted, these are the genes that were actually showing some association, significant association with ROP, and the ones which are referred in black are the ones which withstood the Bonferroni correction. And if the most interesting part was that among the genes that we found associated, these complement factor H and complement factor B, which are involved in alternate complement component pathway, were showing very high association. And the reason we were very intrigued by these two genes was because these are the genes which have been shown to be the major genes for AMD, which is another retinal vascular disease. Now, in the babies, the complements are formed normally. They're supposed to be, they're known to be formed only after 2 years of the age, because all the immunity is provided by the mother. And it was definitely not clear to us that what are these complement genes doing here. So as a matter of fact, we first looked for these complements level in the plasma samples and the serum samples from these babies. We do not see any significant changes in the plasma complement levels. But then as a next step, we wanted to look at what happens in the eye. And the only sample that we could get from the eye was the vitreous humor, which is collected at the time when the vitrectomy is done for reducing the tension on the eye. And so we collect this vitreous fluid, which is removed from the these babies' eyes at the time of the surgery. And since vitreous is actually a tissue which is between retina and lens, it is believed that the secretome of retina can be seen in the vitreous, and that's how that can serve as an ideal system for understanding changes which are happening in the retina. So we then looked for these complements levels in the vitreous of ROP patients' eyes as compared to controls. Now the controls here were the babies who were operated for congenital cataract conditions, and they do a partial vitrectomy, so that's the only possible age match control that we could get. So, and when we compared, we found again there is significant higher expression of complement factor H and also complement 3 in the patients as compared to the controls. So this definitely suggested that there is a significant involvement of alternate complement pathway in the pathogenesis of ROP. Now, to further investigate, we also did the Western blotting to confirm that these find. And in the Western blotting, we found this was done for complement C3. When in the blot, other than the intact complement protein, we also were seeing some of the other smaller fragments of complement C3, which were seen in patients and which were not seen at all in the controls. So that means when a complement protein is activated, it actually leads, its proteolytic cleavage happens, which leads to complement C3A, C3B and C. So what we are seeing here are the proteolytic cleaved fragments of complement C3 in the cases, and which is not seen in the controls. So that means not just the increased expression, there is increased complement activation which is being seen in ROP cases and not in the control. To further confirm this, we actually conducted the global proteomic analysis of the vitreous. I'm not going to show the entire data of the vitreous proteome, but so here again we found there was a different, significant difference among the proteins that were identified in ROP vitreous as compared to control. But interestingly, when we looked at the different complement fragments, we see that again first, when we look at different complement proteins, yes, there is difference in the level expression of these complement proteins in patients as compared to control, and this is the complement C3 fragments. So again, as you can see, there were more peptides related to complement, different fragments of activated fragments of complement component C3 in the patients as compared to control. So this definitely confirmed that there is increased activation of complement pathway in ROP. Now, what causes that activation of complement? So from again going back from AMD, we found that in AMD there is the microglia or the macrophages which actually contribute to the activation of complement pathway. So we, okay, this is just so we alongside complements, we also looked for the anti-angiogenic proteins, and they were down-regulated in patients as compared to control. So we looked for the macrophages in the vitreous samples. So these are the vitreous, macrophages and neutrophils which were seen in the vitreous of ROP patient and which were not detected in the control. And when we use suitable antibody to stain them, they were showing the presence of CD68, which is a marker of activated macrophages or microglia. So now activated macrophages or microglia, they normally can be pro-inflammatory or anti-inflammatory. So to look for their secretome, we actually, we then analyze the presence of the inflammatory proteins in the vitreous samples. And again, we found there was increased expression of MMP9 in the vitreous of ROP patients. Now, MMP9 is known to be secreted by these macrophages, and it actually degrades the small leucine-rich proteins which are present in the basal membrane, and this possibly can explain the vitreous hemorrhage which is seen in these eyes. So we further confirm this by doing the staining of these fibrovascular membranes that we collect. So there was a study where they have shown that the presence of glial cells in the fibrovascular membrane. So we also looked for the presence of MMP9 in these fibrovascular membranes that we've collected from these ROP eyes, and there was, we do not have any control, obviously, because, but in the these fibrovascular membrane, we could see very strong expression of MMP9, and the another protein which is present richly in the vitreous and in this membrane, which is called opticin, it didn't show any expression. Now, opticin is an anti-angiogenic protein. So based on this, we then thought that there could be that MMP9 is known to act on opticin. So there might be some involvement of this role of this opticin degradation by MMPs, and that might be contributing to ROP. I'll explain that in my subsequent slide. Now, all this work so far, when we looked at the proteins in the vitreous, was done in the severe ROP, because that's when we can collect the vitreous samples. Now, how do we now, if we really now want to use this as a marker for risk prediction or for diagnostic, you can't use it. So can we now, and as I said, in the serum and plasma, you do not see these complement levels showing any kind of change. So then we thought of using tear samples as a substitute, and whether, there was two advantages. One, we can collect tears from different grades of ROP, and then we can see whether these proteins that are showing in the vitreous, can they also, do they have a stage dependent change? And second thing, if so, then tear can act as a substitute for doing the biomarker studies. And with this in the mind, we actually looked for tear MMPs and complement levels. So in this, if you can see, tear MMP levels for MMP2 didn't really show much change, but tear MMP9 definitely showed significant higher expression. And now the way we had done this, the tear samples were collected at birth. So when we go for the first screening, at that point of time, we collect tear samples, and then these babies were followed clinically for the development of ROP. So eventually, those babies who had mild ROP at the time of our first screening and then progressed to severe ROP, they had significant higher expression, which is very, very close to the severe ROP cases, whereas those who actually regressed to the, in which the disease regressed, did not show that severe, that higher expression of MMP. So that means MMP9 in tears can reliably predict the onset of ROP. This was done in few samples only. So we then, so this paper was published, and then we extended this study in a larger cohort to check whether red tears can be used for measuring the intraocular inflammation in the ROPIs. And this again, when we did it in larger cohort, the finding remained the same, which indicates that babies who develop, who are mild ROP at the time of primary screening, if they progress to severe ROP, they have their very high levels of MMP right in the beginning. So that means measuring the intraocular inflammation by using MMP9 as a marker can actually serve as a reliable marker for ROP progression. Then we, as I also mentioned to you, that we found that when there is high expression of MMP, opticin expression was almost negligible in these membranes, and we proposed therefore that maybe MMPs are degrading these opticin, and that's how opticin, which is an anti-angiogenic protein, is no more there, and that contributes to increased angiogenesis, which is seen in the ROPIs. So to look for that, we further did some Western blotting and in some more samples. So this is the zymography for MMP. So you see increased MMP activation in the patients as compared to control. And the same patients, when you look for the opticin expression is down. This is done by Western blotting. So then we did an in vitro assay where we used microglial cells, exposed them to hypoxia. So these are the control cells in an untreated condition. They show very high expression of MMP9 and opticin. Under the, when we expose them to cobalt chloride, which induces a kind of hypoxia, the MMP expression increases substantially, whereas opticin expression suddenly goes down. Now, when we treat these cells with doxycycline, which is an inhibitor of MMP activity, or EDTA, which again is a broad inhibitor for MMP activity, the MMP level goes down and opticin levels come back to the. So that definitely clearly shows that elevated levels of MMP degrades opticin, and that's how they are involved in contributing to ROP pathogenesis. So we further looked at the different signaling pathways that in these cells, and this we found that besides MMP, when we inhibit MMP activity, we also see increased levels of these DKK, ERK1, ERK2, which are the signaling pathway involved in blood vessel formation. So these data we have published, and there's another story which I am not talking in detail because of lack of time. So because we said there is increased activation of microglia and they secrete pro-inflammatory cytokines. To further confirm it, we developed certain biomaterials which are basically PLGA, but we have the design that the way, if you see, they were randomly oriented fibers, and these are the fibers which are oriented in a particular fashion. So we found that when you place these cells on these different biomaterial, the cells, they change their phenotype from an pro-inflammatory to an anti-inflammatory type, and this can act as a probably good drug delivery subcarrier molecule. So we are further working on this, and I'll share these data maybe in my next talk sometimes. So now till now we have just done I've shown you the study which we have done more focused on role of complements. Now here we look for the other possible pathways which are involved in ROP, and as Bikash mentioned or Shubho explained, this ROP is not a disease that follow any Mendelian inheritance pattern, so there is not just one gene. So we are looking for multiple gene interactions, and so we chose this approach where we are identifying pathways involved in ROP by using these multi-omics approaches. So the first thing that we did, it was to do a whole exome sequencing. For this, we actually selected the twin cohort of ROP. So we have collected twins where both the twins develop mild ROP, or both twins have either severe ROP or aggressive posterior ROP, and then the both twins who do not develop any form of ROP. So these are who share the genetic component, but and then there was twins where one develop ROP, another one doesn't develop ROP. So they do not, they will show us the non-shared genetic effects. And then comparing the genetic profile, we may be able to identify novel genes and pathways which are involved in, and then this data was further correlated with the global transcriptome analysis that we carried out, and proteome and the metabolome data. So this was the flow for the exome sequencing. So we have analyzed our exome sequencing data by two approaches. In one algorithm, we looked at the targeted genes, which are using a particular algorithm, and another one where we just look at all the variants that were identified by exome sequencing, and then we have identified these genes. So by doing this approach, we identified about 36 rare pathogenic novel variants in about 20 to 20 targeted genes. And then by using the other approach where we have taken, we didn't go by any targeted screening based approach. So there we identified 84 novel genes for severe ROP and 11 genes for aggressive posterior ROP. Now these genes are being validated by the Sanger sequencing and other targeted screening method, and also by using specific in vitro assays. And these were the major pathways that we identified using these genes. And so the notable ones were included fatty acid, carbohydrate metabolism, immunity, metalloendopeptidases, which were significantly. So this confirms the previous finding that we found. But interestingly, now we see fatty acid and carbohydrate metabolism also playing a role. Then this is the data that we generated from the global transcriptome analysis. Now for here we had different stages of ROPs compared with both the premature controls who do not develop ROP, and also the babies of similar age who were born with the mature, with the normal term, and then the data was compared. And obviously severe ROP showed a very distinct profile, and these are the pathways that were again identified using this approach. So notable among them are obviously WNT Wnt signaling, immune, inflammation, metabolism, MAP kinase, fatty acids, which were shown associated. So these actually very nicely correlate with the data that we have obtained from genomics. And then some of these pathways were actually confirmed by doing qPCR for the genes involved in this pathway, and that confirmed these association of these pathways. Like here we have looked at the genes which are involved in the Wnt signaling leading to angiogenesis, and they show very significant association. For example, EGR2, then the FOSB, and then the NOS3. These are also involved in hypoxia signaling and leading to angiogenesis. And then we looked at the genes which are involved in inflammation, and so in NF-kappa B pathway, and again these also in the fatty acid metabolism. So these are the genes which we have validated in an extended cohort, and they show significant association. And this is the data that we obtained from the, I'm not going into details of the metabolomic data because it is still being analyzed, but the initial screenshots show that there is again a very characteristic signature of metabolites in ROP as compared to control. And as you can see from the PCA plot, the cases form a very distinctive group compared to the controls. And these are some of the pathways which are validated by MSMS, and this include nucleotide degradation metabolism, energy metabolism, and amino acid and lipid metabolism. Now, lipid metabolism is definitely very, very interesting, because if you know, omega-3 fatty acid supplementation is given to the expecting mothers, and it was proposed that it can actually help in the prevention of ROP, but when the trial was done, there were variable results. So we were quite interested in looking at first at the fatty acid metabolism pathway. So we actually look for the arachidonic acid, because this is one of the major pathway in the retina. And so there are three major genes which are enzymes which are involved in this pathway. We include CYPs, LOX, and COX. So, and again, when we looked at the expression of these three fatty enzymes, their expression was altered in ROP as compared to control. But most interestingly, alongside, we also looked for soluble epoxide hydroxylase, which plays a major role in the metabolism of lipid oxides and in the conversions of EETs into diols. And so here, this is just a summary of the data that we have done so far. So there is increased expression of CYPs and CYP1V1 and CYP2C, and then there is they convert arachidonic acid into HET, so we do see increased expression of HET. Now, EETs are further converted by soluble epoxide hydroxylase into DHET, and we see a down regulation of DHETs, and a down regulation of DHET would lead to angiogenesis, and which happens via down regulation of the Notch signaling. So we actually have the ones we are showing green and red. They show up regulation, down regulation, which is confirmed by gene at the gene expression level. So we did find this pathway to be implicated in ROP, though we are doing further in vitro assays to confirm this. And so to summarize, based on the omics-based approaches, these are the major pathways that have been identified, and they seem to correlate, these omics approach seems to correlate each other, and that was very interesting. And we are now validating these pathways using suitable assays, and before we really conclude them to be involved in ROP. So that's about this. So this is possible crosstalk that we think is happening, that under hypoxia, basically they leads to upregulation of various genes that are involved in inflammation, and also in the enzymes that are involved in the fatty acid metabolism, and that contributes to inflammation and angiogenesis. This is all from my end, and this is my group. So the work that I have shown today is largely done by this grad student in my lab, Satish Kumar Patnaik, and also the metabolomics work is being done by this guy, Saurabh Kumar. This is my team, and I'm thankful to all the funding agencies for supporting our research, and also to all our collaborators. Thank you so much.
So thank you. I know we ran over by 10 minutes, so if you have questions, we can spill over to lunch, or we can take a couple of questions. But before we do that, I wanted to thank the Vision Research Symposium for sponsoring this, and also Pediatrics Department. Let's thank also Ina and Jenny for putting this together. They really worked hard, reaching out to everyone, all the groups, to do this. Thank you, Ina.
We do have some Zoom questions.
Oh, okay, so we can take them.
We have one for the first speaker, so why don't you guys sort out the second one, and then I'll.
Let's do the second one first.
Yes.
Yeah. So you have the questions, Ina? Do you want to share?
I have one right now, so if there's any people here asking
Yeah,
questions.
so let's go with the questions from the audience. I know Ismail will have a lot of questions, but yeah.
Well, one question is, you identified really the major genes that we already,
Know
they're
about.
very well established to play great roles in the development of ROP.
Yes.
What's your approach in terms of prioritizing which one you want to go after?
Yeah, so that's why we are right now. As I said, we only did a functional validation for complement genes, but there are several new genes, and also my interest is also in the fatty acid genes that are involved in that, and we are actually doing the functional validations for those genes, and then maybe we should be able to answer this. But yes, there are most of these genes, in fact, if you see, are common across other retinal vascular diseases such as AMD and diabetes. So that's very interesting that these diseases span different age group, and yet we see similar molecules for these conditions. So that's something that we are looking at.
Yeah, it's interesting.
Yeah.
I think we had a couple of questions, yeah.
So my question was, with actually regard to diabetic retinopathy,
Mhm.
do you take the same approach of doing the mass spec on the pictures? And do you find similar things between diabetic retinopathy and retinopathy prematurity?
Yeah, so we have done vitreous proteomic profiling for diabetic retinopathy, but though we see some gene shared proteins, but not all are. So there are differences, and in terms of ROP, and but it is interesting that some of these proteins that we have identified in diabetic retinopathy are actually also shown in AMD. So that is something. But in terms of ROP, other than complement and fatty acids, probably there are a few other which I have not really discussed here, but those are the ones which are different in ROP and DR.
And then we had one more here. Katie? Yeah.
So my question was, in the data you showed for the mapping for the ROP, you showed increase in VEGF.
Right.
That's been correlated with increase in KCNJ2, the potassium channel, in diabetic retinopathy. Do you also see that in the ROP patients, where they have this increase in KCNJ2 closely linked to the increase in VEGF?
Yeah, so that's what I said to begin with. If you look at all these retinal vascular conditions, see in all these diseases you see increased expression of VEGF, and that's what is being targeted clinically, right, by giving anti-VEGF, whether it's diabetic retinopathy or AMD, and now in ROP. But I guess there are other molecules which are above VEGF. VEGF is only the result of it. And that's where we have to really, we are looking, our approach is to look for those molecules which lead to VEGF, excessive VEGF accumulation, and not necessarily on the VEGF.
Yeah, Ina, we can have the Zoom question.
So we have one question for Dr. Carl here. It's from Janice Ells, and she says, can you please address treatment, and are you aware of the red light photobiomodulation research? If not, she can share the links, the caption link.
Yeah.
Yeah, I have seen that research. I'm not really looking at it, but treatment currently for ROP is vitrectomy or lasers, but and then on some places they are giving anti-VEGF. We are very conserved in terms of anti-VEGF usage, but that's all about it, and that's why I feel that all those treatments are only once you develop plus ROP or the severe ROP, but as I'm repeating myself, so I think we need to look at targets which can work before the progression of ROP, and that will help to solve this problem largely.
So then the other question is for this is from Dr. Terry Young, and she is asking, how are you now clinically phenotyping your patients' subjects given your findings?
So currently we are still working out on the genotyping, on the genotype profiling. So these are actually classical cases which have already been enrolled. So we have a well-defined criteria to enroll the classical cases which do not have any other secondary complications in terms of secondary glaucoma or any other secondary ocular features. So we have been very critical in classifying those, and currently our approach is to understand the genomic basis of those patients, and then if we are able to show certain signatures across these groups, or if there are multiple different signatures, then go back to them again and then kind of reclassify them. For instance, with respect to PHNR, for instance, we are now getting some different profiles within the same classical category, right? So, I mean, these are very intrinsic or very subtle phenotypic characteristics. And the main difficulty particularly is that we are unable to capture many of the clinical characteristics except for the corneal diameter presenting IOP, and because the eyes are, I mean, compared to an US patients, normally what has been seen is that they present a lot of corneal haze or edema. You would not see that kind of ophthalmos or large eyes here. So that's one of the differences, and so because of that we are unable to kind of see the disc, and so the cup to disc ratio what we showed, I mean Meha would agree, I mean it is difficult to kind of capture for those patients. I mean it's only post surgery when the corneal haze clears out, that is where we are able to do. So but with these kind of parameters like the ERG parameters, so we are trying to see whether we find a different pattern, and whether based on that then there might be a reclassification based on the genetic profiling.
Yeah, that makes sense.
Yeah.
No more questions.
Okay, so then I'd like to thank both Dr. Kaur and then Dr. Chakravarti for, for visiting us.
Thank you.
You know, for their time. And then, yeah, we are looking forward to collaborations and then working together. Thank you for visiting.
And
Thank you all.
hosting. Yeah, thanks for the opportunity.